Hi, this is Dr. Claire, and today we're going to be talking about population genetics and the Hardy-Weinberg Equilibrium. So, remembering back to our definition of evolution, evolution is the change in the genetic composition of a population over time. Now, when we're talking about evolution, we're always talking about populations, because individuals do not evolve. Only populations evolve. So, what is a population exactly? So, a population is a group of individuals of the same species that kind of live in the same area and interbreed. So that's what we're talking about when we talk about a population. In order for a population to evolve, you have to have the change in the genetic composition. So there has to be some sort of variation within that population in order for evolution to occur. Because if everybody in the population was genetically identical, then there's nothing to change into. So you can't have genetic change. So you have to have some sort of heritable genetic variation. A heritable trait is one that is at least partly determined by the genes that an individual has. So that brings us to the study of these heritable genetic traits, which is the study of population genetics. And in population genetics, we have three major goals. We are explaining the patterns and organization of that genetic variation within the population. We're trying to explain the origins of this variation and the maintenance of the variation within the population, and then understand the mechanisms through which this variation may change over time. Okay, so that's what we're looking at. So let's take a look at a population, an example population. So here's an example population. We have a whole bunch of individuals. Each one of the individuals here is represented by one of these little ovals, and the genotype of each individual is listed within the oval. So in this population, we have 30 individuals. Each one has 2 alleles at the X gene. And we have some individuals that are homozygous, which are circled right now, so they have 2 of the same allele. We have some individuals that are heterozygous. We have 2, that means they have 2 different alleles, and there's actually 3 alleles present at this particular gene. So what we need to do is we need to have a way to talk about these genes within the population and look at whether they are changing, okay? So that brings us to the Hardy-Weinberg Equilibrium. Now, we defined evolution as change in the genetic composition of a population over time, or another way of saying that is change in the allele frequencies of a population over time. So what we're interested in is these allele frequencies. And in order to see if they're changing, we need to know what is happening when there's no evolution. So we need to know what happens to these alleles and genotype frequencies if there's no evolution. And if you think about it, if evolution is change in the allele frequencies, then in the absence of evolution, there should be no change, right? And so what the Hardy-Weinberg equilibrium is it allows us to predict what's happening in the population if there's nothing interesting going on, if there's no change and nothing is being influenced, okay? So that's what we're going to be talking about here, is what you expect to have happen when nothing interesting is going on. And then you can compare what you actually see in the field to that model based on nothing interesting happening. And if what you get from the field is different from that model of nothing interesting, then you know something interesting is happening, and then you can figure out what that something interesting is, okay? So this basically gives us what you might call a null hypothesis. It gives us the predicted state when there's nothing happening, all right? So let's take a look at a population and what we're talking about here. So there's several different assumptions that we make about the population when it's in Hardy-Weinberg equilibrium. First of all, we assume that it is a large population, and actually we assume that it's infinitely large. Now in the real world, there's no such thing as an infinitely large population, but a large population of a few thousand individuals suffices, it works. So that's our first assumption. Then we also assume that there's no immigration or emigration. So that means that individuals from one population are not moving to another population. They're not going back and forth, okay? We also assume that there's no mutation, so there's no new alleles arising in the population that we don't know about. We assume that all individuals are mating randomly with other individuals, and we assume that there's no selections. What no selection means is that each individual who is born is equally likely to survive, to reproduce, and has the same number of babies that then go into the next generation. So what that looks like in a basic life cycle is, let's start with the adults. You have a bunch of adults. They pair at random within the population. So each individual randomly pairs with some other individual. They all have the same number of babies. they mate, they produce zygotes. Those zygotes all have an equal chance of surviving to be juveniles. Each of the juveniles has an equal chance of surviving to be adults. And we're talking about an infinite population, and those are our assumptions, okay? Do you think these assumptions are true in the real world? No. I mean, in the real world, there's always mutation. You never have an infinitely large population, and you almost always have some sort of selection going on of one form or another. So in the real world, these aren't necessarily met, but this gives us the model in which to build our predictions about what is happening when there's nothing interesting going on. So these are all the interesting things that could go on, and we're going to build a model of when nothing interesting is going on, okay? All right, so here's our population. So we're going to look at a gene pool. So we have this gene, A, and it has 2 alleles. All right. Now, we need to know some stuff about the alleles in the population. In particular, we need to know what's called the allele frequencies. So we're going to designate the allele frequency of the big A allele as P, and the allele frequency of the little a allele as Q. Now, it doesn't really matter what P and Q are. P and Q there could be 50% big A and 50% little a in the gene pool. It doesn't have to be 50-50. We could have 70% of the alleles in the gene pool are big A and only 30% of the little a alleles, of the alleles in the pool are little a, right? So one thing that should always be true, though, is that when you add up all the possible alleles in the population, we're looking at a gene with only 2 alleles. But if you add up all the possible alleles in the population, when you add them up, they will always equal 1. And the reason for that is that you have a 100% chance of being an allele, basically, right? So you're either an A allele, a big A allele, or you're a little a allele. And if you add up the chance of being a big A allele and the chance of being a little a allele, there aren't any other options in this particular scenario, so they have to equal 1. Okay, does that make sense? Hopefully it does. All right, so frequency of the big A allele, we call it P. Frequency of the little a allele, we call it Q. Now remember, from our rules of probability, frequencies and probabilities are interchangeable. So if you're going to draw out of this gene pool at random one allele, the probability of that allele being a big A allele is equal to the frequency of the big A allele, and we just defined that as P. So that's going to come in useful, okay? All right, so we've got our gene pool. So what's the probability of drawing gametes out of this gene pool and creating a big A individual? All right. So in order to become a big A individual, you have to get a big A from your mom and a big A from your dad, right? And then you become a big A individual. Okay. So what's the probability of getting a big A from your mom? Well, we defined the probability of being a big A as same as the frequency of having a big A in the population, and that we defined as lowercase p, so that's little p, right? So the probability that your first allele is big A is lowercase p. What's the probability of the second allele, the one you get from your dad, being big A, also p. So the probability of getting a big A from your mom and a big A from your dad, you'll notice I said and. Think of your rules of probability. And should lead you to the multiplication rule. So you do the probability of big A times the probability of big A, which is P times P, otherwise known as P squared, okay? So P squared is the probability of any individual that's formed out of the population having the big A genotype, okay? Now if you think about the little a genotype, hopefully it's intuitive that that's going to be, you're going to calculate that the same way. So big A, the probability of having big A would be P squared. The probability of having little a would be Q squared, right? And remember, because probabilities and frequencies are interchangeable, if the probability of being big A is P squared, that means that the frequency of that genotype within the population should also be P squared. Same goes for Q squared, okay? Okay. Now, let's consider a slightly more interesting scenario. Let's look at a heterozygote. All right, so what's the probability of becoming a big A, little a heterozygote? Well, there's 2 ways to get there. So you can either get a big A from your mom and a little a from your dad, or you can get a little a from your mom and a big A from your dad. So did you hear what I just said? I said big A and little a, or little a and big A. Did I do that right? I think I did. Okay, so let's start with the first one. You get big A and little a, right? So you would multiply the frequency of the big A, which is P, times the frequency of the little a, which is Q, right? So big A from dad, little a from mom, PQ. Now, let's say you get the little a from dad and the big A from mom. That's Q times P, right? That's the same thing as P times Q. Doesn't matter which way you do it. And either way, you end up being a heterozygote. So you can either get it, you be PQ or QP. So with the or there, that's the addition rule. We're going to add those two numbers together. And so the total frequency of the heterozygote in the population should be 2PQ, right? Because there's two ways to get there. Each one has a probability of PQ. And so you add them together, 2PQ, okay? All right, so that's how we calculate these equilibrium probabilities, okay? All right, so the frequency of big A, P squared, frequency of little a, Q squared, frequency of the heterozygote, 2PQ. Are there any other possible genotypes with only two alleles at the gene? No, there aren't. So 100% of individuals are either big A, little a, or little a. So when you add those three probabilities together, you will always get 1. If you calculate your genotype frequencies and you add them together and they don't equal 1, you've done something wrong with your math somewhere in there. And you need to go back and check your math and see what you did wrong. So P squared plus 2PQ plus Q squared will always equal 1. If it doesn't, something's wrong, okay? And not wrong with the population, wrong with your math. So go check your math, okay? Okay, so, and this equation here, which is the Hardy-Weinberg equation, you may have heard about before, and it's very useful for, again, predicting what will happen when nothing interesting is going on, okay? All right, so the nice thing about this is that it allows us to predict what genotypes will be in a population, and that is a very useful thing, so we can see if something interesting is happening. And the other thing that's really interesting is that the allele frequencies do not change over time. So they stay constant, and they occur in these. They lead to these predictions in how many of each type of genotype you're going to see. Okay. All right, so I'm going to go through an example of this in the other video for today, so check that out for an example of how we actually apply Hardy-Weinberg equilibrium to check and see if a particular population is in Hardy-Weinberg equilibrium. So check that out. Now, in the real world, populations are almost never actually in Hardy-Weinberg equilibrium, but these predicted genotype frequencies that we get from Hardy-Weinberg equilibrium allows us to tell if something else is going on. So it's basically like a null model, and it's really useful for looking at actual populations, okay? Now, what are some of these interesting things that could be happening? These interesting things that could be happening are mechanisms of evolution, which we'll talk more about on Friday. So those mechanisms of evolution are mutation, gene flow, which is movement of individuals between populations, genetic drift, which is random change due to small population size, natural selection, which is selection, and non-random mating. Non-random mating means that you have preferences, basically, in who you mate with. Non-random mating does not always cause evolution, but it does always disrupt Hardy-Weinberg equilibrium genotype frequencies. So what we're going to do next is look at some examples of these predicted genotype frequencies and observed genotype frequencies, and that can allow you to see if something interesting is going on. So check the next video for that example.